All right, so we're going to start with going through some of the importance and the science behind why it's important for producers to monitor the nutrition of their livestock and some of the tools that you can use for that. Specifically, the tool that we're going to be talking the most about is our tool, the NIRS and NUTBAL System. Then we'll walk you through sampling and mailing. And then using NUTBAL Online to enter your sample information. And then we'll go through how to interpret your NUTBAL results and report. And I'll give you an application of how to use NUTBAL for a specific goal. So starting with the nutritional monitoring of livestock, there are a lot of, animals have varying nutritional requirements that change based on their physiological state or their performance or production levels. So if they're lactating or pregnant or growing, but they all have a base maintenance nutritional requirement that meets their essential requirements for their body to function. But again, those will increase from that base level depending on where they are in lactation, where they are in growing, or if they're working a lot, whether they're working to get to a watering source, draft work, or doing fiber production. That's going to be more related to our sheep and our goats. Okay. Sorry. But identifying and addressing these nutritional needs of your herd and where they're at physiologically, that will help influence, or that's going to influence your body condition of that group of animals in the future. And it'll help you to avoid excessive weight loss during periods of low forage quality or availability. So a time that might be really relevant is during winter months if you're trying to maintain an animal's condition while the primary forage is dormant, or if you're trying to meet weight gain goals in a stocker operation or bringing up a cow to condition for breeding. And then that condition will then impact animal health, reproduction, feed conversion to gain, and all of these factors ultimately go into the producer costs and profits. So an example from scientific literature, or really this is from a review of scientific literature from over 1,000 beef cows, is that cows at body condition scores of 5 and 6 are shown to be superior in cow-calf performance, and that they're more likely to get bred. They have a decreased calving interval, higher weaning weight, and then ultimately higher income per cow exposed. Another thing that you can do if you're monitoring the nutrition of your livestock and your forage is you're able to detect early warning signs for changes in your forage quality. So for instance, the onset of drought can quickly lead to decreased forage quality and availability. And so these things can happen suddenly and quickly impact animal body condition, which will reduce your herd performance unexpectedly. And then forage quality monitoring can provide you the information necessary to kind of catch that as forage quality begins to decline and help you to make decisions in regards to nutrition and performance goals when faced with the onset of potential drought. And then in addition to times of drought, forage quality also just varies from year to year, so that animals, the diet that they consume at one point in one year might not represent what the animals consume at that next time in the following year. For instance, we have some data, and that's actually data that we've collected through fecal samples from forage consumed by cattle in Wyoming in 2015 and 2016. And so we're showing quality in terms of crude protein, so that's one of the nutrients that we analyzed for here. And the crude protein average over time in all of Wyoming for 2015 is in blue. And you can see that crude protein might peak in 1 month during a year, so it looks really high here in about April and May, but it's much lower in the next year, so 2016 is represented by the red line. But nevertheless, with yearly nutritional monitoring, producers can start to develop a general idea and the trends of forage quality and how your herd responds to these changes. Your herd will respond to these changes, and that can be useful for long-term planning. Taking all of these factors into consideration, there's a lot of variability in the way livestock nutrition can be addressed. This figure displays the various expenses associated with cow-calf operations, and no matter what group of profitability the production was in, the feed and the grazing are the highest costs, but that also offers the largest areas to improve. Monitoring livestock nutrition in a grazing study poses a specific set of challenges. So for free-ranging livestock, it can be hard to estimate forage quality because animals have free choice of grazing various forage species. And then in extensive systems, plant nutrient composition and overall plant species composition will vary with changes in terrain, climate, and time of year. And then on top of that, grazing animals can even further selectively consume certain parts of the plant, given that there is sufficient forage availability and diversity for them to be that selective. And that will make the diet different than what you might find if you just went and you did a forage clipping and clipped all of the forage in one forage ring or plot. And so the reason that the GAN Lab exists is to provide livestock producers with effective solutions to monitor the nutrition of their animals, primarily those grazing forages, so that when the forage quality is known, producers can implement grazing management practices and supplemental feeding strategies to optimize animal performance and feeding costs. So to aid in this decision-making process, Texas A&M University, in collaboration with the NRCS and other universities and organizations, have developed tools and techniques to improve livestock nutrition management. One technology that allows us to rapidly determine nutritional characteristics of a grazing animal diet is fecal NIRS, or fecal near-infrared reflectance spectroscopy, and that's what we use here at the Grazing Land Animal Nutrition Lab. So NIRS is a technology that examines the interactions between matter and electromagnetic radiation in the near-infrared wavelength. And that's a lot of big words, but hopefully I'll be able to break it down in a way that makes some sense. So here's the electromagnetic spectrum, and NIR wavelengths are located, or they represent the portion of the spectrum that is just longer than the visible wavelength. So they are not visible to our eye, but they still interact with matter the same way visible wavelengths interact with matter. So for instance, with human color vision, the property makeup of an object affects whether or not certain light waves are reflected or become absorbed. And with visible light, the color wave that's reflected is what we see. So in this instance, we see red on a rose because red is the wavelength that is reflected. With near-infrared reflectance, it's a similar concept. So a light source is used to illuminate a material, so in our case it's fecal matter, and the near-infrared wavelengths are either absorbed or reflected from that illuminated material. And that absorbance or reflectance is based on the different chemical properties that the light interacts with. The wavelengths are reflected at different strengths and absorbed at different strengths, and that level of absorbance and reflectance can be recorded by instruments which are connected to our computers, and that is saved to us as a unique NIRS spectrum that represents the chemical composition of that sample. So for fecal NIRS, when we started developing this technology, it started with cattle on a forage-based diet plus a fecal sample. We collected the fecal samples and produced an NIR spectra, which is the same as what we do every day now with y'all's samples when they come in. But we add in the information of the forage diet. So we collected samples on that forage diet and did nutritional wet chemistry analysis to determine the nutrients by the standard traditional protein and energy methods. So we knew the nutrition in terms of protein and energy of those diets. For fecal NIRS, we essentially pair the fecal sample with the known nutrient composition of those diets, and we're able to match the spectra with those nutrients in a way that we have an equation that was developed to predict where these different peaks and troughs fall in the spectra and how those relate to the different nutrients in the diet. So we created this equation. So now all we need is a fecal sample that we can produce an NIR spectra from, and that is converted through this equation into the diet quality of what the animal is consuming when they produce that fecal sample. So to use in combination with the diet quality predictions supplied by fecal NIRS, the GAN lab developed NUTBAL, which is a nutrient balance simulation model. That is computer software, basically, and that evaluates nutritional well-being of livestock. What NUTBAL essentially does is translate the diet quality information from the fecal samples into performance outputs, such as average daily loss or gain, and it evaluates how current nutrition influences body condition and performance over the next 30 days. That can then be incorporated into management practices to design optimum supplemental grazing and feeding strategies to meet herd performance goals. In a few minutes, we'll start the NUTBAL online tutorial and go through in depth what client-supplied data is required by the NUTBAL model. And again, these inputs from you will be based specifically to your sample, so they'll be herd characteristics, pasture conditions, any supplemental feeds that were offered, and these are used to parameterize our model and provide performance information that is specific to the herd that corresponds with the fecal sample. And these inputs from the producers on our sample information sheets or our NUTBAL online sample submission, they're really key to providing the most accurate assessment of your local conditions, your diet quality, and animal performance. So this is a really big and complex looking diagram, but we tried to kind of put together what goes into the NUTBAL model so that it can best predict the pasture conditions and animal performance. And so this right-hand, there's my cursor, this right-hand research and technology development box describes the different types of data that make up the foundation of NUTBAL. So in order for NUTBAL to evaluate a wide variety of grazing animal scenarios, it's been outfitted with equations that take into account differences in nutritional requirements according to factors that might influence those requirements. So cattle breed, size, physiological state, body condition, and activity levels, those will all impact their energy and protein requirements. And much of this information has been derived from the National Research Council for Beef Cattle Nutritional Requirements, a publication that they produce that's essentially kind of like the beef or the cattle nutrition Bible, basically, that's used by cattle nutritionists, feed companies to formulate feeds. And it's also the standard for beef cattle researchers, nutritional researchers. And then additionally, the NIRS technology development and calibration trials that were done at the beginning that paired those fecal samples with the known diets, and then they've been done throughout to sort of keep up with our equations and keep those equations calibrated correctly. Those keep the NIRS predictions as accurate as possible. On the left side box, that represents the client supply data that comes with the sample. and those come through the sample information sheets, the physical ones that we provide with the boxes, or you can submit it online. And these inputs are specific to each sample and the animals that are represented by that sample. And they allow NUTBAL to use its built-in data and prediction equations to characterize performance potential of that specific herd based on breed potential, management practices, and location information, which NUTBAL actually uses to pull climate data corresponding to that sample date and the address that's provided for your properties. And we'll talk about that again later in the walkthrough section. So once we at the GAN lab process and scan your sample, that diet quality information is integrated into the model, sort of like the last piece of a puzzle, and that allows the model to function and produce its end product, that end product being the predicted herd performance over the last 30 days. And so NUTBAL, or at the GAN lab, we have two service options that you can receive. So our standard report will include that NIRS report of your forage quality and the animal performance projections over the next 30 days. And then you could also upgrade to an advisory, and that includes the standard report, a breakdown explanation of the standard report, and a supplemental feeding recommendation. So you could either provide us with a supplemental feed that y'all have available to you, or we'll, if you don't do that, for samples that don't do that, we try and select one that's regionally available, or provide a couple different options of how to meet your animal's requirements through supplemental feeding. So to put it all together and kind of show what goes on after your sample arrives, it's unboxed out of the cooler, and we assign it a GAN lab number so we can track that fecal sample all the way through all of our processes. It's emptied into drying trays and placed in an oven to dry for 36 hours. That dried sample is ground and homogenized. And then it's scanned with an NIR spectrophotometer, which actually takes multiple sort of images or pictures, spectral pictures, and then as it rotates that sample around, so it captures that sample from a lot of different angles and points and gets a representation of the species that you provided us. So once we have the forage quality information, we run it through our performance model, and then the results are sent back to y'all, the clients. And then this is just a note on invoicing and payment. It's kind of like our last step. Once we get those back to you, the results back to you is when we actually will send you an invoice. And we ask that you don't send the checks with the samples, as they can sometimes get wet or ruined by leaking samples. So after this presentation, if you would like to see additional resources describing the applications of fecal NIRS, or need help determining what to enter in your sample information, or just to work through NUTBAL online, please check out our website. And this is what the page looks like. If you Google just CNRIT TAMU or TAMU GAN Lab, TAMU being Texas A&M University, it'll come up. And there are some older NUTBAL tutorials and information sheets available, as well as several tools and documents to help you determine things for your parameters on our sample sheet. So we have tools to help determine body condition, scoring, or if you're having trouble estimating forage cover. And these are found in our publications and tools tabs. We also have a direct link to our NUTBAL online website, which is a different website from this, but you can get there easily from here. And then I don't have it on here, but we have, there is also a link here to our YouTube channel where we have previous webinars available, as well as shorter videos broken up. So like the specific shorter tutorials for walking you through sampling or submitting a sample, or and then this webinar will actually be broken down and end up on our YouTube channel. as well as our Facebook page. The videos are also available on our Facebook page for you to access at any time.